Yeah, welcome to Rare Connection, where we connect rare disease patients, educate medical professionals and the public, and help shine a light on research and clinical trials and advancing the future of medicine. Today, we're exploring an area of medicine that combines cutting-edge diagnostics, targeted therapies, and hope for patients facing some of the most challenging cancers affecting the central nervous system. Joining me today is Dr. Marc Hedrick, President and CEO of Plus Therapeutics, a clinical stage pharmaceutical company focused on developing targeted radiotherapeutics for difficult to treat cancers, including leptomeningeal metastases and recurrent glioblastomas. Before entering the biotechnology industry, Dr. Hedrick trained as a general vascular plastic surgeon and served as an associate professor of surgery and pediatrics at UCLA. He also co-directed UCLA's laboratory and regenerative bioengineering and repair before moving into leadership roles in regenerative medicine and biotechnology. Today we'll discuss the challenges of diagnosing and treating cancers and the spread of central nervous system, emerging radiotherapeutics approaches, the roles of advancing diagnostics and like CNSide, and what these innovations could mean for patients and families. Dr. Hedrick, welcome to Rare Connection.
Thank you.
You trained as a surgeon and spent time in academic medicine at UCLA. What originally drew you to medicine?
Actually, I wanted to take care of patients with rare diseases. And that started with wanting to become a pediatric surgeon. And by definition, almost anything that involves pediatric patients is rare. And I was really interested in the surgical end of that. And then that led to fetal surgery, which is operating on the baby before the baby's born while it's still in the womb. And many things develop prenatally such that when the baby's born, the effects of the disease are already locked in. And in many cases, fatality is the norm with those. So interacting super early in the womb, mid-gestation, you can, with the baby or the fetus safe inside the mom, then you can correct the developmental abnormality. The patient's born and would survive and thrive where they otherwise wouldn't. So that became ultimately a career in translational medicine, which is taking new therapies, new drugs, now diagnostics, and then translating them into people. And then now I'm on the commercial end, which is, okay, let's don't just get them into the clinic. Let's actually get them to approval and actually helping people and helping them. You can actually increase your reach, or one can increase their reach by getting new therapies into the market. So, all patients theoretically can take advantage of them.
So, what was a particular experience as a surgeon or researcher that sparked your interest in developing new therapies?
Well, when you see the patients every day in your clinic, and you see the ravages of the disease, and in some cases for pediatric conditions, the disease effects are already locked in by the time that they would come to me as a pediatric surgeon or a craniofacial surgeon. And particularly with craniofacial surgery, there's a big issue with scarring, particularly around the face and the head and the neck. Whereas in the fetal environment, the fetuses heal without scarring. They heal through a regenerative process. And so, for example, babies with cleft lip, we designed a way to treat them while they're still in utero in the mom's abdomen, with the idea that then they could, when they're born, the cleft lip is already fixed and the scarring is mitigated. Same thing with spina bifida, or something called diaphragmatic hernia, which is typically fatal if not treated before birth. So it's sort of seeing the effect in the kids when they're born, and then recognizing really you had to go backward in time to treat them to be able to ensure that they get the best possible outcome. So that really drove my interest in doing fetal surgery. But that's sort of super and ultra rare. And now we're taking care of patients that have central nervous system cancers, which are still orphan diseases or rare diseases, but they affect a much wider population of patients and are the worst of the worst cancers. They're the cancers that have had the least improvement in survival of any sort of anatomic group of cancers that are out there. And in our view, the biggest unmet medical need, but still affects, on a relative basis, a rare population of patients.
Mhm. So how has your perspective evolved over the course of your career?
Well, initially, I I had one tool in my bag effectively, which was a scalpel, and so that the span of the things I could offer patients were at the tip of the scalpel. And as I've gone throughout my career, I recognize that the scalpel's limiting, and there are other approaches to take care of patients: pharmaceutical, therapeutic, medical device, as well as surgical procedures. So right now, what I'm doing is sort of a combination of all of the above. While there's what we do now is it's part surgical, part therapeutic, part diagnostic, and part pharmaceutical. So we're a technology agnostic company, and we think that that's for central nervous system cancers, which, by the way, the survival in those has been largely flat for 50 years, despite MRI, despite all the advances in healthcare that we've seen that have increased all other cancer survival about 300%, still relatively flat with CNS oncology. And so we felt like bringing to bear an all of the above approach is the only way we can make meaningful improvement in survival. So it really builds off what I was doing early in my career to a much more expanded view of the world in terms of the toolbox, but our focus is limited to the central nervous system.
So what ultimately led you from treating individual patients in the operating room to leading companies focused on developing therapies for thousands of patients?
Well, it's a bit of an extension of what I mentioned before, which is, uh, as a surgeon in the operating room with your anesthesiologist and your team there, you're still limited by what you can do with two hands. And I realized through my research career and then into the business career that we can make a much greater impact and accelerate that impact if we can build out the team, bring on people with other talents that can bring their two hands and their brain to the table, and we can basically invent things that then we can make broadly commercially available, train other doctors and providers in how to use those, and not just treat the one patient that I'm treating, back when I'm a surgeon, but we can treat the entire community that's affected by that disease. So the amount of leverage we get off of what we do now has the potential to be much greater than what an individual surgeon can do in the OR. Even developing new procedures, you still are limited by what other doctors will do in the operating room, or whether they will adopt or not adopt the procedure. And then bringing in now advanced data analytics, which is sort of a whole new area for us, but we're implementing very rapidly. We can stitch together everything and bring that to bear, not just to an individual patient, but to the community.
So how does your clinical background influence the decision you make as a CEO of Plus Therapeutics?
Well, it's in one way, that's a really simple answer. It's because we think like providers, we think like physicians. And I'm as CEO, that ethos rolls all the way down the company. We face every decision we make. As part of that decision, we're thinking about the patient. What's in the patient's best interest? And we stay very close to the patients. I run in 10Ks with some of our patients after they've been treated with glioblastoma. I'm at times in the OR. I'm in nuclear medicine with our patients. We try to stay very well connected. Here we are in our home office in Houston. We're a half mile from MD Anderson near the Texas Medical Center, which is the largest medical center in the world. So we stay very closely connected as part of that background. And I found that serves us very well in every time we have a decision to make. The patient's always sitting on our shoulder with a seat at the table, because that's the way we think.
So many of our listeners may not be familiar with leptomeningeal metastases. What exactly is it?
So there's an epidemic of brain cancer in the US, and it's driven by leptomeningeal cancer or leptomeningeal metastases. So it's a increasingly common, although technically still rare. It's sort of pushing the boundaries of what constitutes a rare disease. But it's a metastatic complication of many very common cancers like breast cancer, lung cancer, melanoma. The drugs for those patients are, in many cases, working well and turning these fatal diseases into chronic diseases. So because of that chronicity, the patients are living longer, but the cancer is not cured. They still have cancer cells in their body, but they're at bay, and the patient is living their life as long as they stay on therapy. But inevitably, those cells that are still there will escape, get into the circulation, pass through the body, and will end up in the brain or the spinal cord. And one of the key areas where they end up is in the protective environment called the cerebrospinal fluid. So the brain and the spinal cord are encased with a fluid layer. And that fluid layer, once it's affected by tumor, is almost impossible to treat, or currently impossible, I put it that way. And the same things that we have as protective mechanisms to protect our brain and spinal cord, protect cancer cells once they get into that environment. So it's a unique challenge. But leptomeningeal cancer is essentially metastases to the fluid space around the brain. We know it affects about 125,000 patients per year in the US, probably affects something closer to 400,000 because it's significantly underdiagnosed. And so that's a big part of the equation in terms of bringing in all of the above approaches. We recognize we have to solve some diagnostic issues as well as therapeutic issues to make an impact on leptomeningeal cancer.
This is one's not on the let's see, what makes them difficult to diagnose?
Well, first of all, the imaging is not great. So even MRI is technically advanced as it is, and as as common as it is as a diagnostic methodology, it's not really great at seeing leptomeningeal disease because it's not a big tumor that you can easily image. It's often can be very occult or small, like little BBs or little like a saran wrap on top of the leptomeningeal membranes, and it can be difficult to see by MRI, difficult to quantitate, and then it becomes difficult to tell whether a treatment is working or not. So the normal imaging, it really does not work well. We're very reliant on getting some of the fluid around the brain and testing it. And that's a major limitation because the standard of care for diagnosing cancer in the fluid around the brain is a technology that's been around well over 100 years. It's called CSF, or cerebrospinal fluid cytology. And that gives the doctor a yes, no, or maybe answer. And oftentimes when it says we can't find cancer, it's actually there. The test is not sensitive enough. So it's very, very poor sensitivity. And so we have acquired recently a diagnostic company that can find one cancer cell in an entire specimen. So it's highly sensitive, highly specific. And then besides confirming the diagnosis very quickly, which is very important in these patients for a variety of reasons, that it can tell us what sort of mutations are there, what sort of drugs the tumor might be susceptible to, and it gives us a quantitative assessment of their tumor burden. And so we can use that as a way to continue to alter the therapy along the way, to optimize it, to try to reduce the tumor burden and improve outcomes. So really the diagnostic part been very critical to finding those patients that are undiagnosed or underdiagnosed, and then match them to a right therapeutic approach, and then monitor their treatment going forward so we know if they're getting better or getting worse. Because oftentimes, by the time you get outward symptoms of they're getting worse, then the game's already over, and we can't wait that long in these patients.
Mm. So, let's see, which cancers which cancers most spread to the
Yeah,
leptomeninges?
theoretically anything. Generally, the more common the cancer, the more likely it is to spread, just because there are more patients with the more common cancers. And so breast cancer counts for the majority or significant minority of the patients that get LM. Lung cancer, common cancer, and a cancer that commonly metastasizes to the leptomeningeal space or the CSF, melanoma, gastrointestinal cancers, prostate cancer. So all of the ones that when you watch TV and you see cancer commercials for drugs to treat breast cancer and other things, those are increasingly metastasizing to the leptomeninges. And then once it gets there, there are no approved treatments. Most drugs don't get into or around the brain, so they're blocked out of the brain. And then treatment becomes very, very difficult. In fact, about half of the patients that have leptomeningeal cancer, the doctors throw up their hands and send them to hospice because there are no good alternatives. Or they were diagnosed so late that they're in a very, very weakened, critical stage, and the patients are really succumb emotionally, mentally, and then ultimately kind of biologically to the cancer. So getting an early diagnosis can keep them in treatment. And we're ambitious enough and perhaps crazy enough to think that we can, if their breast cancers become a chronic disease, once it gets into the leptomeninges, we think we can turn that into a chronic disease as well. Perhaps we can even cure patients with the right kinds of drugs or therapeutic approaches. So but the upstream part of their therapy begins with an accurate, rapid, assessment of whether they have it, what it's susceptible to, and then a way to monitor and manage them so that we can iterate their therapy along the way to continue to give them a high quality of life and a longer life.
What symptoms might patients experience when cancer is spread into the cerebral spinal fluid?
So because that fluid space, it's a really good question, because that fluid space is contiguous with the entirety of the brain and spinal cord. So from literally from the top of the head all the way down to the lower end of the spinal cord, you have both the central nervous system and the peripheral nervous system. So anything from sort of the neck up to the neck down can be affected. So you can have things that are very, very discrete and pinpoint. For example, a facial nerve weakness, or a eyelid that isn't working properly, or a hearing deficit, or a weak arm or leg. So something that's very obvious, very, very testable, very discrete, or it can be very vague, or could be something such as a headache, or a sort of a vague sensory deficit in my arms. I'm just not feeling in my fingers like I usually do, or the side of my face feels a little funny. And those kinds of things can, for a variety of reasons, can come and go in patients. But the key thing is, if the patient has uh, a cancer or an advanced cancer and develops a neurologic finding, then they should be evaluated for leptomeningeal cancer or metastases to the CNS. And that workup would include typically imaging, CAT scan, MRI, a neurologic exam, and then a lumbar puncture. And so basically the information you get from a lumbar puncture today, cytology, protein, glucose, are very sort of standard things that have been done since the early 1900s, and they're not particularly sensitive or specific. Whereas we have a complete diagnostic portfolio that we can run on the CSF from a lumbar puncture and define the disease, define a treatment path, and then monitor the patient. So the key thing is a patient with advanced cancer that has a new neurologic finding absolutely needs to be assessed and probably imaged and should have a lumbar puncture.
Mhm. So, what's the current prognosis for patients diagnosed with leptomeningeal metastases?
So the best way to think about it is in a bifurcated way. So the patients that are seen on a more advanced uh, set of symptoms or disease, and they're not seen by a specialist or someone that sees this type of thing a lot, they oftentimes will go to hospice and simply live a few weeks, maybe 2 to 4 weeks, something like that. So they really get supportive and end of life care. Uh, the patients that ultimately are seen, not so late, or are seen and assessed rapidly, and a treatment regime is established, those patients can live a few months, call it 2 to 5 months. That's still unacceptable. That's the best we have,
Yeah.
but in our view, that's still unacceptable. Our lead drug is called Reyobiq. It's a targeted radiotherapeutic for patients with leptomeningeal cancer. And in the phase one, we showed that we could extend life on average to 9 months. And we think there were a number of patients that lived very significantly longer than that 9 months. So we think that there's a lot of upside therapeutically to improve survival, to improve outcomes, improve patients' neurologic status and functional status with early intervention with drugs that are appropriate for this particular disease. So while survival is very poor now, I think we can see over the horizon here that we can make substantial gains in survival and improve outcomes.
Let's see. What are the biggest unmet needs facing patients today?
Well, I think I'll just focus on central nervous system oncology patients. So
Mhm.
it starts with diagnosis, uh, and so confirming a tissue diagnosis early is very important. And so catching the symptoms early is very important. That's number one. Number two is finding treatment strategies that cross the blood-brain barrier. And I've alluded to this, but I haven't really talked about this. The big problem with CNS oncology is that barrier that is in place in all of us to keep poisons and bad things out of our brain and spinal cord, to protect our, our essentially our most, probably our two most important organs are our heart and our brain. And protecting our brain is largely handled by the blood-brain barrier, which is a biologic barrier that keeps poisons and things out, but it also keeps about 98% of drugs out. So most drugs don't get to the brain. They get everywhere else, but not to the brain. So having therapeutic strategies that will remove the blood-brain barrier from the equation and allow the therapeutic to get to the brain, spinal cord, the leptomeninges, where it can do its thing. So having therapeutic strategies that will solve for that become critical. And then finally, ways to monitor therapy. It's a very blunt instrument to do a neurologic exam and try to tell whether the patient's worse, the same, or better. But having something like our CNSide assay. CNSide is our diagnostic company, and it's a central nervous system diagnostic platform that assesses multiple different diagnostic endpoints. Can actually tell quantitatively whether the cancer's the same, better, or getting worse, and we can titrate the therapy to that data. And it's shown also to be prognostic. The higher the cell count, higher the tumor cell count in the brain, spinal cord, the more advanced the disease is, and the worse the prognosis is. So we have something quantitative to shoot for, and I think that solving for the blood-brain barrier and solving for the early diagnostic understanding of the patient's condition and monitoring are the 3 to 4 things that are most important in these patients.
So you mentioned CNS. Okay, so we're getting to CNSide diagnostics now. You already mentioned what it was. So, how is cerebral spinal fluid collected, and what information can physicians learn from it? So I think you mentioned that one too.
Well, kind of. I alluded to, but yeah, let me answer that kind of more
Okay.
directly. So the typical way a patient is that has neurologic symptoms, uh, a normal part of the workup, the clinical workup of that, is a lumbar puncture, which is some local anesthesia, tiny needle, generally in the lower back, patient sitting up in an outpatient setting. And it's a very simple procedure to go in, sample some fluid, put it in tubes, send it off, and get a diagnosis. So a patient, for example, that has advanced cancer that develops a new neurologic symptom that's suspicious will get a lumbar puncture in that way. That's sort of the standard way of getting it. However, when patients are diagnosed with leptomeningeal cancer and they're being treated, they get something called an Ommaya reservoir. That's like a cancer port that is put in the bloodstream, except this one's not put in the bloodstream. It's put into the fluid around the brain. Just a little, very simple surgical procedure. It's a rubber membrane that sits just under the skin of the scalp and has a small pigtail catheter that goes into the ventricle or the cistern, where it's easy to get the fluid out of it. And for those patients, that their fluid can be sampled in a couple of minutes in the clinic as an outpatient with essentially no anesthesia. So it provides real-time, uh, long-term access to the cerebral spinal fluid, not only for for diagnosis, treatment monitoring, but our lead drug, Reyobiq, is actually for leptomeningeal cancer, is instilled through that port. So it provides two-way access, cells and fluid coming out for diagnostic analysis and treatments going in, and gives kind of real-time access to that protected space. And it takes the blood-brain barrier out of the equation. So it parachutes the drug beyond the blood-brain barrier and gets it to where it's needed, to what we call the region of interest.
So, what is it? What is detecting tumor cells in in cerebral spinal fluid so important?
So normally in the cerebral spinal fluid, that fluid is turned over very rapidly, about 5 times a day. So the body's making fluid and the body's reabsorbing it. And there's a normal cell turnover that happens as part of that process, as the regenerative cells are renewing the leptomeningeal membrane and the other very highly specific cells of the central nervous system that are there. And there's typically no blood there. There's really not many things other than just the slough cells and the actual liquid. And the liquid provides almost a structural barrier-like insulation for trauma. If we fall and hit our head, that fluid helps protect the brain. However, in a disease state, depending on the disease, you'll find different things in the fluid. If there's a bleed in the brain or a hematoma, or trauma, you can find blood cells there. That's not normal. If there is cancer in either the brain tissue itself or in the fluid around the brain, which is called leptomeningeal cancer, you can find sloughed cancer cells. And that's absolutely not normal. You shouldn't have a single cancer cell in that space. So finding, needle in the haystack, a single cancer cell in a big fluid specimen, that's not good. That tells you have the disease. So you could also detect tumor DNA. The problem with tumor DNA is that it's not a cancer cell. It means there's cancer somewhere, but you don't have the cell. And having the cell is really, in our view, it's the critical thing to have. We do biopsies all the time in patients to confirm whether they have a tumor. And the reason we do that is because we want to see the cell. We want to assess the DNA of the cell. We want to assess the markers on the outside of the cell. And the only way to do that is with what we call with tissue or with cells. And so having access to those cells and being able to interrogate them, in other words, ask questions about what tumor markers are they expressing and what drugs are the tumor susceptible to, and is a tumor cell going up or count going up or down? All those things are important to the doctor to manage the patient. So CNSide addresses all of those issues at once with a single lumbar puncture.
So, how does CNSide differ from traditional methods to diagnose leptomeningeal cancer metastases?
Yeah, I mentioned earlier something called cytology. Cytology is just a general term of looking at cells from the body tissue. That could be from the skin, from the eye, from the internal organs, and it's a very sort of rudimentary early 20th century, late 19th century technology where you rub a tissue or a specimen on a glass slide, kind of smear it across, and put very standard dyes onto the slide that will differentially stain the cells that are there. And then the way it's been used for central nervous system cancers in the lumbar puncture and CSF fluid is to see if the cells look like they may be cancers or not, which is a very surprisingly subjective analysis. And that's why you can't get a quantitative assessment because you're just smearing things on a slide that doesn't really tell you how many tumor cells that are there. You can't quantitate them, in other words. But it it's different in every laboratory because this is done down in in every hospital. Pretty much every hospital does cytology or pap smears, for example, so that happens in the cervix. So it's the same core technology. And so it gives you it gives one sort of a yes, no, or maybe they have it, maybe they don't. Maybe we should repeat it in a couple of weeks. It gives you a very subjective sort of answer. Now, in 1904, when this hit the market, that was new technology back then, and it was the best thing that we had. But today is very different. And so we've developed a cell capture technology. It's based on a microfluidic chip. We run the entirety of the fluid from the CSF specimen through the chip, has a variety of antibodies and other chemical mechanisms that trap only tumor cells. We can read a variety of different tumor cells, and we can trap any of those cells onto the chip. And then we can stain that chip with a number of different things to bring out the cancer-related markers, gene expression, protein expression, and really anything you can interrogate chemically, we can assess for on the chip. So we can assess the entirety of the cells that come from the brain fluid on a chip, and then we can take that data, feed it to the computers, and give an analysis back to the doctor that really will give them a guide to diagnosis, treatment monitoring, and prognosis.
So I've read that CNSide can provide quantitative cell analysis and molecular characterization of tumor cells. What does that mean in everyday language for patients?
So the first thing it does is in essentially 4 days, we get the specimen, and in 24 hours, we know if they have cancer or not. We can get that information back to the doctor. And then in the next 2 or 3 days after additional testing, and then validation and going through and double checking, have a pathologist look at it and so forth, just to make sure all of our computer-based analytics are validated by a real doctor. Then we send that back to the patient. So in 4 days, the doctor can have an answer on whether they have a cancer, and then what a treatment path would look like. And the doctor that can then make very quick decisions for the patient. Once they know they have cancer, talk to the patient, decide whether treatment's appropriate. If it is, they can put an Ommaya reservoir in, get that on the schedule, get that in very quickly. And then as soon as that's in, they can start treating the patient. So you can take process for the patient that could take 2 to 4 weeks to figure out everything. You could do it in really a few days. And we see this all the time in our therapeutic trials, that patients that take a while to get into therapy, they can decline very rapidly with leptomeningeal cancer because it is in that protective environment. Drugs don't get in there. It's like very, very rapidly growing weeds and that are deadly. And so compressing that time to diagnosis and treatment from weeks to a day or two can mean the difference between a patient going to hospice or going into treatment and that treatment being ineffective to having an effective treatment, and they can live months and we think ultimately years with LM. But getting all of that kind of crammed into that first few days is really critical for these patients. And the flip side of that is for a patient that has advanced cancer, that has neurologic symptoms, we can tell them very definitively, hey, you don't have leptomeningeal cancer. Good news for you. In fact, that's probably the majority of the patients that come in. There's suspicion of LM, but they don't have it. Their neurologic symptoms are from something else. We can rule that out. And both the patient, the doctor, and the family all have a big sigh of relief. And we don't have to put a Ommaya reservoir in and treat them speculatively. We can treat them on real hard, rapidly obtained data. and then rule out those patients that don't need treatment.
So can the test help physicians determine whether treatments are working?
Very, very, very important point. So the cytology, as I mentioned, the current gold standard is a yes, no, or maybe. And whether the cytology becomes, if it's positive and becomes negative, that's a very blunt assessment of whether the patient might be getting better with treatment. MRI is not really quantitative in this sense, so it again gives one a very qualitative, subjective answer about response to treatment. Changes in neurologic exam or symptoms can be spurious, late, difficult to quantitate. So that's no way really of sensitive way following patients. But CNSide gives the patient and the doctor a tumor number. For example, we can say you have 5 tumor cells per milliliter of fluid, or you have 5,000 stem cell or tumor cells per milliliter of fluid, or you've gone from 50 to 25, or 5,000 to 100. That data is very germane, quantitative, reproducible, and actionable from the doctor's perspective. So it's on one hand, we could, with standard of care, it's subjective, kind of iffy. It's a lot of more guesswork. We can bring these patients into the 21st century and tell them very specifically, do you have it? Is it getting better? Is it getting worse? Oftentimes, as we know from cancer therapeutics, cancers can respond to a drug and then they can quit responding, and the tumor, whether they're in remission, they can actually get worse. And then we can pick that up very quickly and make a change. The other thing that's very relevant to this particular question that you asked is there's something called genetic drift in breast cancer patients, lung cancer patients. And what I mean by that is when a patient is diagnosed with breast cancer, they'll have a tissue biopsy, and the doctor will assess things like HER2, their estrogen receptors, and be able to tell the patient and doctor whether they're receptive to different kinds of chemotherapeutics. However, that biologic assessment, their receptor status, their HER2 status, for example, can change from the breast into the brain. So one really needs cells from the brain to really assess that, because that could be very different than the primary tumor. And it can switch. There's something called the flip, where you can flip from one receptor status to another one over time, as these tumors can be they're escape artists in a way, and can be very adept at resolving ways to not be susceptible to chemotherapies. We can pick that up in real time. We can do CNSide once a week or once every 2 weeks, so we can get very dynamic look at how that tumor is responding to treatment, whether it's trying to escape from the current therapy, and then we can recommend different treatments. So it becomes a very dynamic, interactive, ongoing testing process to try to find the right therapeutic approach. And as I think I mentioned much earlier, a new and important emerging part of the business is advanced data analytics, or AI. So having the opportunity to have a tremendous amount of longitudinal data on a particular patient and submitting it to the type of analysis that only this kind of advanced data analytics could do is already opening up new ways to categorize, prognosticate, and treat and follow these patients in a way that we really probably couldn't do a few years ago, because those data analytic techniques weren't available.
Do you see advanced diagnostics becoming increasingly important in precision medicine?
it will be for us. I can tell you about how we think about the world. We think about the world in terms of diagnostics, therapeutics, and advanced data analytics. So we give it an equal seat at the table with the therapeutic approaches and the diagnostic approaches. And one of the things that we're trying to do by focusing on CNS oncology, which is unusual to have companies that do diagnostics and therapeutics, is the advanced data analytics becomes the glue between those two parts of the businesses and interconnects them and help us drive a pipeline of new therapies and new diagnostic approaches that we probably wouldn't otherwise have access to. So we view it as essential to what we're trying to do. And we think we're, I guess, arrogant enough to think that we, as a company, can actually start bending the survival curve for patients with CNS oncology and cancers by virtue of melding those three parts of the business together.
Many people are familiar with radiation therapy, but may not know what radiotherapeutics are. What are radiotherapeutics?
So they're drugs that deliver radiation, and contrast those to the way radiation has historically been delivered for decades, which is through an external beams. So in other words, you have a radiation source outside of the patient. That beam is collimated, focused, and the attempt of the doctor, the radiation therapist, is to treat the tumor and only the tumor. Well, that's impossible because it's going through normal skin, skull, brain, and then out the other side. So you're treating other things. And so it's very limited. But as it relates to cancer of the brain, it is the best treatment. So if you look at all treatments that are out there today for brain or CNS cancers, the most incremental benefit that one sees is with radiation. And that's why it's typically given in these patients. Even if they're getting a drug or surgery or something, radiation is almost always given. It's an essential part of therapy. Well, radiotherapeutics is now a way to deliver radiation, but with a drug. And that, rather than being photon radiation, which is what external beam radiation does, which is a kind of radiation, this is nuclear radiation. So, for example, our drug is actually a combination of three different components. And one of the components is a salt of aluminum that's put into a nuclear reactor and becomes irradiated, and then it becomes a source of beta particles, which are high energy electrons that are unstable, that get sent out and then kill rapidly growing tumors. So it's a drug. It's made in a nuclear reactor. It's delivered in such a way to take the blood-brain barrier out of the equation, and then it delivers all of its radiation to the tumor or the area around the tumor that's at risk for local growth, and then burns itself out, and then ultimately it is processed and excreted by the body. So, for example, for glioblastoma, we can deliver up to about 750 gray, probably more, but that's the maximum we've done. So radiation absorption is measured in gray. So with our drug, we deliver, call it 750 gray of radiation, typically with external beam. In the same group of patients we treat, they get about 35 gray. So we're essentially 10 to 20 times the amount of radiation, and we're giving it in a single shot. Whereas with external beam radiation for glioblastoma, it's typically given over 4 to 6 weeks in very small increments, because if you give it all at once, it can be catastrophic for the patient. So we can give our targeted radiotherapeutic in a concentrated way, deliver all of the radiation load in a single treatment to the patient, 10 to 20 times the amount of the radiation, with essentially no systemic toxicity. So it really is, in our view, a game changer, for these patients. And then it leverages the thing that I mentioned before, that is radiation is the best killer of CNS cancer. The key is you just have to get enough of it there. And then the more you get, the better it is. And so we've proved that. We have a recent paper in Nature Communications showing, to our knowledge, the highest amount of radiation ever given therapeutically to patients. And then we looked at how much radiation is enough, and we never got to a maximum dose. So in other words, for every incremental amount of radiation we give the patient more, the patients do better and live longer. So more radiation is better, we're finding. And so the key is to find safe ways to target it to these patients.
How do radiotherapeutics differ from traditional external beam radiation? We answered that one.
Yeah, I think we hit
Yeah,
that. Yeah.
is okay. So, why are radiation therapeutics generating so much excitement in oncology right now?
So it's kind of, we're kind of in the second wave of excitement for radiotherapeutics. If one knows much about healthcare, you probably have heard of thyroid cancer and radioactive iodine. And there's a very specific way that iodine, which is essential to good thyroid health, gets taken up. And by delivering radioactive iodine that gets concentrated in the thyroid, that is a principal way to treat both cancerous and non-cancerous disease of the thyroid. And that's been around since the first half of the 20th century. And iodine is very similar to the radioisotope that we use. So why has that come back? Because now there's a lot more radioisotopes that are now known, and there's chemistry and biology behind them and radiobiology that show that they work. There are increasing number of ways to target them, to attach them to radioisotope drug conjugates that will concentrate the radiation into particular types of tumors or organs. And there are new novel delivery mechanisms that allow that to happen. So it's a combination of all of those things that have caused a renewed interest in these, and that renewed interest is developed over the last 10 years or so. And now with a few examples, now those drugs are working and working very well. And neuroendocrine tumors, in prostate cancer. And I think ultimately, most tumors, there will be a radiotherapeutic option because it massively exceeds the amount of radiation one can deliver up and above external beam that's been used for 100 years. So it's really exciting time, and radiotherapeutics will populate all areas of cancer treatment.
So, what advantages do targeted radiotherapeutics potentially offer for conventional treatments over conventional treatments?
Well, so there's a couple of different ways to answer that. One is that it fits very seamlessly in the overall cancer treatment paradigm, which is to incorporate surgery, radiation, and drugs, pharmaceuticals, chemotherapeutics, all together in a way that combines to improve outcomes. And that's essentially at a very kind of non-specific way the current standard of care. And but in many cases, surgery is not an option, or the cancer is advanced or spread, can't be surgically removed. Maybe it can be debulked, but not cured. And then so chemotherapy and radiation become a part of the mix. To the degree the disease is systemic, in other words, it's spread widely, surgery's off the table, radiation may be off the table or largely off the table, and you're really relying on pharmaceuticals that can circulate throughout the body and that can get there. So in one way, it fits very normally into that sort of three-tiered approach. I think where it kind of begins to perhaps stand out a bit is because it combines elements of all three of those approaches. Radiation can ablate tumors and sort of kill everything, which is sort of a way to, so kind of what you try to do with surgery is to take it all out and, put it in a pan and take it out of the body. Radiation can, in a way, can kind of do that. Radiation can be, as a drug, radiation therapeutics can be delivered systemically, so it can circulate. And then with appropriate targeting mechanism, it can be concentrated in the organs or the tumor types that preferentially take that radiotherapeutic up, so it functions like a drug. And then ultimately, the kick that it delivers is the radiation. It's what we call double-stranded DNA breaks. So the way ultimately the radiation kills the cancer is it irreparably damages the DNA of the tumor cells, so that the tumor can't work around that. It's simply apoptosis or dies. And so that mechanism of action is very clear, very standard. Everybody knows how radiation works. And so breaking the DNA such that it can't be repaired is the way the radiation takes that energy and uses it to kill cancer. So it can actually perform the radiotherapeutic across all three of those different parts of the therapeutic stool.
Mm-hmm. So what makes cancers of the brain and central nervous system particularly challenging when it comes to drug delivery?
Yeah, I alluded to this earlier. The blood-brain barrier is a problem. So as I mentioned, only about 2% of drugs will normally get through the blood-brain barrier. Oftentimes in cancer, the blood-brain barrier is locally disturbed and it becomes a bit more porous, but that's an exception, not the rule. And because the brain doesn't have the normal containment mechanisms that other cancers have, it tends to infiltrate. So there's a blood-brain barrier you have to worry about, but you have to worry about local infiltration. CNS cancers that start in the brain don't tend to metastasize. They tend to take their horrific detrimental effect to the patient by infiltrating. And so, and the brain is an organ that is so mission critical, tissue preservation and normal brain health. We have to be critically observant to protect that at all costs. So it becomes a very high stakes game of trying to be incredibly aggressive at killing the tumor and being incredibly protective of the normal brain tissue that's needed to function. So it's that high stakes game that we're playing. And so managing around the blood-brain barrier, managing around the infiltrative nature of these tumors, finding things that will deliver their payload to the tumor and only the tumor, and minimize the amount of those payloads that kill the cancer are often toxic to the brain. And so minimizing that effect outside the tumor becomes a very critical balance that we're trying to achieve. So that just makes it just functionally so difficult to treat these patients, and is a reason why, despite billions and billions of dollars of investments over the last 50 years in trying to treat cancers of the central nervous system, the survival curves are largely unchanged for 50 years. And so that's why we decided as a company to throw away that 50-year-old playbook and try to take an all of the above approach, or to put it a bit more technically, take a technology agnostic approach to the field. And we're open to anything that works, or to the synthesis of bringing multiple things together at once. For example, bringing diagnostics, radiotherapeutics, and advanced data analytics all together in a single company focused on one thing, is extending life in these patients. We think has the best chance of materially improving survival in these patients.
Mhm. So can you explain the concept of treatment directly to the tumor or affected area rather than throughout the entire body?
Yeah, there are some ways that have been shown to either disrupt or bypass the blood-brain barrier. One such thing is called high-frequency ultrasound, which can be delivered and temporarily disrupts the blood-brain barrier and allows its permeability and allows stuff to get in that wouldn't otherwise get in. That's an example of a way to bypass the blood-brain barrier. We've adopted two technologies that are already in the clinic, well proven, that can work depending on where the tumor is. So to take a step back, we think about brain cancers in two ways for the purposes of delivery. So one is the cancers of the brain tissue itself. That's called the brain parenchyma, the brain substance. And there's a technology that came out of the NIH, I think maybe 30 years ago, called convection enhanced delivery. And it basically requires that in a minimally invasive way, up to five, in our case, five different catheters are put through very tiny drill holes. You have to go through the skull. The catheters are put in very precise geospatial arrangement based on the tumor and a variety of other things that are become very technical. Those catheters are put in, they're secured into place, and then the drug is infused through those catheters in a very strategic way with the goal of encompassing the entirety of the tumor and the normal brain that's at risk for the tumor spreading. As I mentioned, that tumor spreads very easily and kills patients locally, not metastatically. So we can get very sophisticated in terms of how we image those tumors, assess the tumor size, location, and the region of interest, and then through convection enhanced delivery, we can treat the entirety with our radiotherapeutic. And oftentimes I'll explain that as it's biological fracking. If we're in Houston, oil and gas industries are very important to this area. Or Bickley gas is pulled out of the ground by pushing water under high pressure to break up shale deposits to free up natural gas. Well, it kind of works the same way. You're pushing the drug in under pressure. It breaks through the connections in and around the tumor in a very predictable way. And then once we turn off the pressure, the drug immediately stops, stays in place, delivers all of its radiation, and then it's normally absorbed after it's completely burned out and the radiation has been delivered to the tumor. We call that biological fracking, also known as convection enhanced delivery. So that treats the parenchymal part of the disease. For tumors of the fluid space, for example, leptomeningeal cancer, I mentioned an Ommaya reservoir, a very simple outpatient surgical procedure, and gives the provider two-way access to the fluid compartment. It can get drugs in and can take fluid out diagnostically. So right now, we're focused on those two device-based delivery methodologies. They're commercially available essentially at any hospital that has a neurosurgeon. They'll have access to both of those tools. And so there's really no limitation in terms of where the patients can be treated. Any hospital in the US has the ability that has a neurosurgeon can do both of those particular delivery methodologies. But there are other things that we're looking at as possible ways to take the blood-brain barrier out of the equation. But those have been really difficult to resolve, haven't been resolved to any great degree in the last 50 years. And so we're looking at that, but right now the money is in those two things that I mentioned before.
Mhm. So okay, so this is getting to Reyobiq. So Plus Therapeutics is developing Reyobiq. What is it and how does it work?
Yeah, Reyobiq is our lead central nervous system cancer targeted radiotherapeutic. It's a complex molecule. We take a salt of aluminum. Uh, it there's some chemical changes that are made. Then it gets irradiated. We have a specific way to grab each molecule of radioactive, now rhenium. Rhenium's a heavy metal. Rhenium in its non-radioactive state, about 80% of rhenium that's used commercially is used in jet engines for propeller blades because it's so heat tolerant. But we turn a different atom into rhenium. Rhenium is grabbed, and then we load it into a lipid-based molecule that holds it in the brain so that once we instill it, either by CED, convection enhanced delivery, or through an Ommaya reservoir, it stays essentially in the space that you put it, delivers all of its radiation, and then goes away. So why do we use it? It's principally the physical properties of the rhenium itself. So with radioisotopes, and this cuts across all different kinds of radioisotopes, there are certain physical properties. For example, how long does it take to decay? In other words, how long is it delivering its radiation? The time to decay with rhenium is ideal. It has about a 90-hour half-life. So once you put it in, it could stay there literally for weeks and deliver its radiation before it's resorbed. So path length is important as well. So when the radioisotope is put into the brain, the high energy electrons that kill the cancer, how far do they go out? It goes out about 4 millimeters, which is right in the sweet spot for the zone of delivery. It's not going out too far, but it's going out far enough that you can get a lot of spatial leverage, if you will, to kill just the tumor and the adjacent tissue that one is targeting. The chemistry of it becomes very important, and rhenium has very ideal chemistry for how we use it and load it. The amount of energy or punch that it provides is right in the sweet spot as well. And then another very key feature of rhenium is it has two kinds of radiation. So the majority of its radiation is in these high energy beta particles that do the DNA breaking. But it also produces a gamma particle, which is a different lower energy particle, but it allows us to image it. So use aspirin as an example. You take an aspirin pill, it goes into your body. You have no idea where it goes, and you hope that your headache gets better, but you can't. There's no way to sort of image it in the body and know that it's getting to the exact spot of your brain or your elbow where the pain is. With rhenium, we can do something called a SPECT CT, which is a radioactive type of CT, and we can through our rhenium radioisotope give the doctor very detailed information about where the drug is at any point in time and how much radiation it's actually delivering to the tumor. So in our paper I mentioned before, we were able to quantitate how much radiation was actually getting to the tumor and then correlate that with improvements in outcome. That's a very powerful, not only clinical tool, but an R&D tool. So every patient, in a way, becomes their own clinical trial. So we know how much radiation we delivered. We know how much is getting to the tumor. We know how long it's there. We can calculate the amount of radiation that the tumor or our region of interest actually got, and we can correlate that with improvements in outcome and so forth. And it allows to really be on a very fast iteration cycle from an R&D perspective to make rapid gains and improve the treatment approach on the fly in a phase 1 to get to where we are now, which is a couple of phase 2s for glioblastoma and for leptomeningeal cancer. And then take that data, go back to the FDA, and then we're starting a pediatric brain cancer trial here very soon based on that amount of very unique delivery data that only a radioisotope like this can provide.
Mhm. So to finish that, can you tell us about the ReSPECT-LM clinical trial?
Yeah, so ReSPECT-LM trial, our Reyobiq trials, their suffix is ReSPECT, and then LM is for leptomeningeal cancer. So as I mentioned, leptomeningeal cancer is known to affect about 125,000-150,000 patients in the US every year, but thought to be 1 or 2 times higher than that, maybe up to about 300,000 to 400,000 patients. So that's for those patients. There's nothing approved for these patients. We have a diagnostic in CNSide that helps us really confirm the diagnosis quickly. They get an Ommaya reservoir, and we've completed the phase 1 ReSPECT-LM. It's told us what the maximum tolerated dose of Reyobiq is in those patients. We showed that on average about a 50% improvement in tumor cell counts in those patients and approximately a doubling of survival in those patients in our phase 1 trial. We're now in call it a phase 1A, 2B trial, and that's to optimize the dosing regime. So we know what the maximum dose is in the proper single dose range. Now we're trying to figure out how often should we give it. And then once we figure that out in this phase 1B, 2A, then we'll be going into a bit of an expansion trial to get additional safety data with our dose and dosing regime. And then we'll go into hopefully an approval trial with the FDA. The FDA has been super supportive of the ReSPECT-LM trial and try to get this to patients. They understand the desperate need for the epidemic of LM. So that trial is now expanding to a number of well-known academic clinical sites and will be pretty spread out regionally throughout the US here in the next couple of months.
Are you currently recruiting for it?
We are.
So what are you looking for in patients?
Yeah, so these are, since we're still technically in the safety phase, we're open to pretty much all tumor types. I'm not just limited to breast cancer or lung cancer, for example. We have to prove that they have LM, and they have to be able to have a Ommaya reservoir in place, and so and then some typical, exclusion criteria in terms of what's called their Karnofsky score, their degree of debilitation. We don't want for this trial patients that are very, very advanced. So patients that are more on the early to moderate sort of area of the disease. But generally, we're open to most patients. And there's a ReSPECT-LM, a clinical trial site that's easy to find. You can Google ReSPECT-LM and find those trial sites and patients can get into referral very quickly for those trials.
Where is it currently at? What locations?
Well, I think the way I think about it is we're pretty much covered all regions in the lower 48 right now.
Uh-huh.
So, or will be very, very soon.
Is there a specific age range you're looking for?
No, LM is actually surprisingly, it tends to affect a younger group of patients. So most of the patients are before they've reached their sort of Medicare age. They're going to be in their advanced, more aggressive cancers in their 30s, 40s, and 50s. But no, there's no to my knowledge off the cuff, I don't think there's an age restriction.
Mhm. So, what have researchers learned so far from studying Reyobiq in patients with leptomeningeal metastases?
Big picture, what we've learned is the brain is very radio tolerant. So we can give extremely high doses of radiation. To my knowledge, we've given the highest that's ever been given in the history of mankind, and the brain is very tolerant to those. So that's one thing. The brain can tolerate very high doses of radiation. Number two is that there's no upper limit thus far that we've reached, that the more radiation, the better, which makes sense. But you have to prove it to make the statement. And I think we've proved that. We've proved that we can reliably deliver those very high doses to a variety of different brain cancers in different locations through convection-enhanced delivery. And we've shown that we can use dosimetry. We can quantitate the amount of radiation given to the tumor. And that's very important. And we've also, I think, very importantly, unlike most targeted radiotherapeutics, by the way, we've shown that we can do, we can use our approach and have essentially no systemic toxicity. So in other words, we don't have to worry about the bone marrow toxicity, liver toxicity, spleen toxicity, other organ toxicity, heart and lung toxicity by the combination of a targeted radiotherapeutic and targeted delivery. So I would say right there, 5 or so things that are kind of general and unique for what we're doing. More specifically, we've shown that Reyobiq, even in phase 1 trials, shows very promising signs of improved survival. Usually in phase 1 trials, you're not looking for survival. You're really looking for safety. But very early in our clinical development, we're showing pretty dramatic improvements in survival in patients where the disease is relatively early, and we can cover the tumor with high doses of radiation. And so I think we're very, we hopefully got to prove it to our satisfaction and the FDA's satisfaction, but we're very encouraged by what we've seen so far from a safety and efficacy perspective for Reyobiq in a variety of different kinds of brain cancers.
Plus Therapeutics is also studying recurrent or glioblastoma. What glioblastoma and why? What is glioblastoma and why has it remained so challenging to treat?
So a glioblastoma in adults, or it's also called high-grade glioma in pediatric patients, is the most common primary tumor of the brain. So we talked about brain metastases, leptomeningeal cancer. Those are much, much more common than glioblastoma, but those are metastatic cancers. Glioblastoma is a primary tumor that starts and stays in the brain. And glioblastoma affects about 15,000 patients per year in the US. So it's 1/10 or 1/20th the number of patients that will get leptomeningeal cancer or parenchymal brain mets. So it's rarer, but it's in its most aggressive form, high-grade glioma or grade 4 glioblastoma. It is the most aggressive, and it grows very rapidly. It tends to be infiltrative in the brain. It's even with a CAT scan or MRI, what you see is not the full extent of the disease. So it tends to go beyond what one can image on standard imaging. So you have to treat a bigger area than one would otherwise think to get meaningful improvements in survival. So in patients that are initially diagnosed, they live approximately on average 15 months. So it's pretty short longevity after diagnosis. But in almost all cases, near 100% of them will recur or will not be adequately treated. And so they will go into a recurrent or inadequately treated group. And in that group of patients, no matter what you do to them, on average, they live about 8 months. So whether they're in a, whether you try this drug or that drug or radiation or no radiation, or put them into a clinical trial, 8 months is about how long they live on the recurrent side. So in our phase 1, called the Respect GBM trial, we showed that we could essentially double survival in those patients where we got more than 100 gray of radiation. So I mentioned absorbed radiations measured in gray. We're giving 3 times the amount that's typically given, and those patients will have a doubling of survival. And then in our phase 2 trial, we showed that in all comers, not the ones that just got 100 gray, all comers, we could get a 60% improvement in survival midway through the trial. So we're showing pretty encouraging signs of improvement in efficacy and relatively safety profiles in those patients. But it's difficult because of the things I mentioned, and then you attack on the blood-brain barrier, the limitations of amount of external beam radiation that one can give these patients, which is essentially inadequate. It gives you some local control, probably does better than anything else, as I mentioned, but just it doesn't give you really meaningful improvements in survival. And that's why the recommendations for the National Comprehensive Cancer Center Network, they recommend that patients with recurrent glioblastoma should go directly into a clinical trial. So it's a really tough group of patients. We're targeting them first because it's essentially 15,000 patients per year. And then I think longer term, Reyobiq could be potentially used in the primary setting. But we're starting with the recurrent patients in our current trial. That phase 2 is almost complete, should be complete relatively soon. We'll analyze the data. We'll talk to the FDA and come up with a plan to do an approval trial.
So are there any encouraging developments emerging from the glioblastoma program?
Well, other than what I've mentioned, I think I covered it. I mean, I think the key thing that is really encouraging to me is that we've learned that the brain can handle super high doses of targeted nuclear radiation, and that the more radiation you give, the better the patients do, the longer they live. That's a very important biological and clinical finding, in my view.
Yep. So I understand that there's also work being done in pediatric brain cancers. Can you tell us about that research?
Yes, and it's an extension of what we've done for glioblastoma in adults, but those sort of translated down to pediatric patients. And these really are classified as sort of rare, ultra-rare diseases. And the 2 tumors that we are going after in our current trial, it's called Respect PBM. It's basically pediatric brain cancer. So we're going after patients that have something called ependymoma and high-grade glioma. High-grade glioma is the pediatric version of glioblastoma, and that represents approximately about 500 to 1,000 patients in the U.S. every year. So very small number of patients, but these poor kids really, there's very minimal things that the doctors have to offer to them. And then ependymoma is not quite as lethal or aggressive as glioblastoma, but it's very difficult to eradicate. And serial recurrences, where they have surgery, radiation, chemo, whatever, tends to come back over repeatedly over many years. But both of them have the common theme that they're both very radiosensitive. So we're going after both of them in a sort of a complicated clinical trial design. So we should be enrolling relatively soon. I think we We can start referring patients now to our lead clinical site, which is in Chicago at Lurie Children's Hospital, which is under the Northwestern academic umbrella. And that's also available if you Google ReSPECT. You'll see the ReSPECT trial for pediatric brain cancer. And we're very, very hopeful that the things that we've seen in adults will translate into the pediatric population.
So what would you like newly diagnosed patients and caregivers to know about leptomeningeal metastases today?
I think that ask for CNSide. Go to their doctor. Not all of these patients that have leptomeningeal cancer are cared for by neuro-oncologists. They're cared for their primary doctor or their medical oncologist that may not be aware that there's such a sensitive, specific, and versatile diagnostic option and we have reimbursement for that. We should have 150 million covered lives for that by the end of the year. We're partnered with big payers like United, Blue Cross Blue Shield. We're getting closer to having Medicare and ultimately Medicaid coverage. So that is largely and increasingly a covered test that's available that can not only nail down the diagnosis if the patient unfortunately has it, or it can rule it out if the patient doesn't have it. We can give them very rapid feedback and, hey, good news, you don't have LM. But then we can very much engage with them in the therapeutic process as their tumor morphs or changes or tries to escape chemo. We can help give information to their provider or their providers that they can respond to that and improve their lifespan and improve their symptoms. So that's the first thing I would do is get a quick diagnosis, and then secondarily get into therapy and ask for CNSide and look for Reyobiq. There are very few clinical trials for LM right now. So we're going to be really expanding our trials here very soon as we start to move into an approval trial. So I think look for both of those things once they have a sense that they might have LM or that they do have it.
Mhm. What questions should patients ask their healthcare team if they suspect central nervous system involvement?
Well, I think the first thing to figure out is that it's getting the diagnostic workup done quickly. And there's something called therapeutic nihilism. This is not a term that I invented, but we hear our neuro-oncologists use that term, therapeutic nihilism. And it commonly occurs in leptomeningeal cancer because doctors will view it as sort of an end-of-life diagnosis and put them into hospice. So I would encourage patients to reject that therapeutic nihilism and say, well, look, come in. What about CNSide? What about this new diagnostic technology? I want to use that and incorporate that in my care plan. And number two is there are trials that are out there and they're emerging in dynamic therapeutic approaches. Some are leveraged the utility of CNSide, but also things like targeted radiotherapeutics. So I think the questions would be, do you take care of a lot of these patients? I want to be aggressively diagnosed, managed, and then treated, referred into clinical trials. So those are the kinds of dialogues I would have with providers if it's suspected. And hopefully the physician will use CNSide to rule it out. But if they have it, then rapid treatment and getting into trials is very important.
How important is early detection when dealing with CNS cancers and metastases?
Well, I'll give you an example. Early on in our phase 1 trial, we had a number of clinical trial sites that were enrolling patients, and the sites who had the best outcomes were the ones that had a very rapid time to diagnosis and then to treatment. And then on the flip side, we found that the patients that were put on maybe a slower track to getting in to get a lumbar puncture, and then having their having the old-fashioned, if you will, tests done that aren't very clear, that had to be repeated, that in the span of a week, 2, 3 weeks, patients can get very demonstrably worse very quickly and end up in that group of patients that no more treatment options are really available to them. So I think we see it in our trials. Doctors tell us this frequently, is very rapid diagnosis and then very rapid inclusion into aggressive treatment protocols is the way to go, because these patients can decline very rapidly.
What role do patient advocacy organizations play in advancing research?
With metastatic cancer, particularly LM, they're the normal societies that support and advocate for patients with brain cancer. But the feeder into LM is through breast cancer, and probably the biggest number of societies and dollars for patient advocacy go through breast cancer for the reasons we all understand. But breast cancer, lung cancer, melanoma, gastrointestinal cancer, patient advocacy groups, all of those are increasingly aware about brain metastases and leptomeningeal cancer. So they have elements that think about that, because it's increasingly important topic for their patient groups. And as I mentioned, and going back to one of my first comments, there's an epidemic of leptomeningeal cancer and brain cancer, because the drugs we're using for those primary diseases are working better and better. And the final common pathway these patients will succumb to their disease is through brain-related metastases.
How can patients learn about clinical trials that may be available to them?
clinicaltrials.gov, or they can look up ReSPECT. ReSPECT trials, and it'll come up very high on a Google search.
So, looking ahead 5 to 10 years, what advances are the most hopeful when in neuro-oncology?
From my perspective, the thing I want to see in 5 to 10 years is I want to see that survival curve improving. We want to take something that's been flat for 50 years and then start to raise it in a meaningful, measurable way. That's really what I'd like to see personally. And that's my personal goal. How we get there, to me, is less important. The more important thing is that we get there. And we have, as a company, are trying something completely different. Let's take an all of the above approach. Let's bring together the experts and diagnostics, therapeutics, advanced data analytics, bring those all to bear to improve survival. So what do I see on the horizon? I think that will make that future state a reality of improved survivals and longevity for these patients. It's ways to bring the blood-brain barrier into submission, and figure out ways that we can bypass that, figure out ways we can get higher and higher amounts of radiation to these tumors, figuring out ways diagnostically to come up with systems and specific measurements to show which patients should get which treatments and which patients are measurably getting better, and having an early warning system to detect the need to change therapeutic regime or to show improvement. And maybe they can reduce therapy in these patients or take them off therapy for LM if we reduce their tumor cell counts to zero. Or using advanced data analytics to figure out, what patient groups, need certain kinds of treatments. So we can take our diagnostics and our therapeutics and focus it on specific patient groups or types of tumors. So I think those are just some ways. There are different kinds of molecules, different kinds of targeting mechanisms. That could be a very long discussion, but I think we're open to what works, and we don't come to the game with a bias. We're all of the above company. Our goal is to improve survival and outcomes.
So, what is one misconception about brain cancers or leptomeningeal metastases that you wish to more people understood?
The one thing I would say is there's hope that this is universally a group of patients that once they get the diagnosis, they tend to lose hope. And so I think there is hope. There's hope in the approaches that we're taking and other companies are taking. And there are things that are happening today, which we think will end up in the market relatively soon and be broadly available to patients. So, don't give up.
Yeah. See, if listeners remember only one thing from today's conversation, what would you like it to be?
Well, I think that if a patient is concerned about this diagnosis, there is a way there's a better way to make these types of diagnoses and give them actionable recommendations for treatment. And that's available today. And very soon, a group of drugs will be available that really will offer, kind of build upon the improvements in survival that we see with better diagnostics, and then start to improve longevity and outcomes.
Mhm. What can people learn more about? Where can people learn more about Plus Therapeutics, CNSide diagnostics, and ongoing clinical trials?
Come to our website, follow us on social media, plustherapeutics.com or the Respect trials, or CNSide, our diagnostic division. And there'll be many more important, germane, and interesting updates coming over the rest of the year.
Mhm. So, thank you, Dr. Hedrick, for joining us today, and for sharing your insights into the challenges of diagnosing and treating cancers of the central nervous system. Today, to our listeners, if you like to learn more about today's topic, please check the links in the show notes. If you're enjoyed this episode, please subscribe to our connection on my YouTube channel, Rare Chef. The video viewers can leave a comment below, or and audio listeners can use the send me a text link in the show notes. You can also follow Rare Connection on Facebook, X, and LinkedIn. I now have switched my Facebook page over to the current account because my account was at one point hacked, and so I had to switch it. And there is also a Facebook group for Rare Connection. Remember that this podcast is for educational purposes only and is not intended to replace professional medical advice. Always consult your healthcare team regarding medical decisions. If you or someone you know is struggling with thoughts of self-harm, call or text 988 in the United States and Canada, and contact your local emergency services or crisis hotline. My next episode will be the third anniversary episode on June 17th. I will be talking with Dr. Greek Goiterstam from Make-A-Wish Foundation. This will also be my 10th international episode. It will be talking to her in Amsterdam. Until next time, stay informed and stay connected. And remember, no one should face a rare condition alone.